Ito na ko.
Ay, nandiyan. Nice na.
Ano?
Bakit mag-visible na yung kan? Yung susunod. Yeah, let's start now. 1, 2, 3, go! Hello, guys. So today we're going to present our virtual lab DNA extraction. Biotechnics virtual lab. Paano ba na-explain ko? So first we have in our virtual lab a image of the DNA. So Excel, Joy Salas, can you read the description of the DNA on the right side?
So, in this experiment, you need to isolate some DNA from a human test subject. Why? You may ask, do you need human DNA? So, press next.
Okay, next. So we have here a genetic testing and body identification and analysis forensic evidence. So yun yung mga purpose bakit need natin i-understand ang DNA, yung forensic identification, forensic evidence, genetic identification, tsaka body identification din nandun kanina. So, ako muna lang magbasa sa description dito. Scientists isolate DNA for a variety of reasons, some of which include genetic testing, body identification, analysis of forensic evidence, and analysis of forensic evidence. So, next slide. Si Rowena, makabasa ka dito sa description? Sa right
Wala
side.
si Ro. Wala
Ay,
siya.
wala si Rowena. Ikaw na lang ulit, Texie. Go.
DNA extraction is typically the first step in a longer laboratory process. DNA extraction is an important part of the process because the DNA first needs to be purified away from proteins and other cellular contaminants. We need cells because that's where the DNA is inside. Almost every cell in our body is a nucleus, and inside each nucleus is about 2 meters of DNA.
2 meters haba. Para ng DNA sa nucleus. Wow! Chromatin. Chromosome pala. Joke lang.
Di ba chromatid pag single lang yung like isang ano lang?
Oo, isa lang.
Sa DNA.
So where do we begin? First, we need to collect some cells from our test subject.
Bira si Rowena.
Ayan si Rowena. Oh no. Matay sa mga niya.
Wow.
Kuha tayo ng cells. Cells galing sa kanyang saliva. Ay, saliva.
Chicks.
Chick cells ba yan? Or naganga siya? Sa bunga nga. Rowena, makabasa ka ng description sa right side. Babasa ako.
Ito, the skin on the inside of our mouths loses thousands of cells every day. These are the cells we're after. Narinig niyo ako?
Narinig na lang.
Ito yun?
Po. Nice ka. So, mag-next ka na tayo. So, kumuha siya tayo ng skin galing sa mouth ng ating sample. Next is, let's get started. The steps you will follow to purify DNA from a chick swab are shown below. Collect chick cells, breast cells open to release DNA, separate DNA from proteins and debris, isolate concentrated DNA. So, next slide. Next part ng virtual lab. Here are the materials and equipment you will need to purify DNA from the chicks. Okay. Natin yung mga materials mo. Open ba yan?
Ano ba yan?
Hindi siya open. Makita niya lang yung mga materials dito. Meron tayong microcentrifuge. May hot bath. Ata to hot bath. Feel ko hot bath. Meron tayong, ano tawag niya ulit? Yung parang dropper. Kalimutan ko. Pipette. Meron tayong pipette. 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 Meron tayong pang swab. Meron tayong container ng sample at saka iba't ibang solution. Mention man niya mamaya ibang solution. So, warm water bath, centrifuge, cubicle swab, micropipetters, sample tubes, and ito na, lysis solution, concentrated salt solution, suspension buffer, ethanol, and ano yung pinaklas dun?
Ewan.
Bilis kasi siya masyado. The suspension, ethanol, at saka last is isopropyl alcohol or rubbing alcohol. And ito na. X mabasa mo?
Oo. So, drag the swab up to the inside of the test subject mouth to collect a sample. So,
Yeah.
i-drag mo ran.
Tinag ko na. Ayun.
So, next. Wow.
Wow.
These
So,
are chicks.
ikaw na sila.
Next, you'll place the swab into an Eppendorf tube. Look closely at the swab. Note that this is covered with hundreds of tiny chick cells. Inside each chick cells is a nucleus, and inside the nucleus is DNA.
Wow, chick cells.
Ikaw na ran.
Okay. So, gumamit siya next ng pipette para ilagay yung lysis solution. Si Ro na. Ro. Mabasa mo, Ro. Ang next step.
Hello?
Ro. Nawala ulit si Rowena. Baka may talaga net.
Hindi na ran
Nandito
siya,
siya.
pero wala nagsasalita.
Okay yung net. Ako na lang siguro muna. The end of the swab must be cut off so you will be able to close the tube. Using the micropipetter, add some lysis solution to the tube. Lysis is a Greek word for means to separate. May sinabi na rin yung meaning dito yung lysis solution. Yung magse-separate ng siguro yung mga debris at saka yung DNA from yung sa sample container natin. Nilagay niya ng lysis solution using a micropipetter. Warm bath. Place the tube into the warm bath.
Warm water bath.
Warm water bath. Sorry lang. Wala nga ng warm water bath. Ay, drag ko pala. Oh, wow! Ito na yung DNA natin with histones. Rowena, nakabasa ka na, Ro? Si Rowena. Ako na lang. The lysis solution you just added contains two important ingredients, detergent and an enzyme called proteinase K. The detergent disrupts the cell membrane and nuclear envelope, causing the cells to burst open and release their DNA. The DNA is still wrapped very tightly around the protein called histones, and the proteinase K cuts apart the histones to free the DNA. So pinaburst niyo yung histones gamit ng lysis solution natin, pati ng detergent para ma-disrupt yung cell membrane. First yung detergent, mag-open yung cell membrane para makapasok sa nucleus natin, pati sa nuclear envelope. Tapos next yung proteinase K, siya yung needed na enzyme para mag-break down yung histones para ma-free yung DNA na naka-roll sa histones.
Funnel.
Tapos, feel ko yung purpose ng warm bath dyan para yung, atag dito, yung temperature, like maging ideal temperature yung para sa enzyme, para mas mabilis yung process niya ng pag-breakdown sa histones. Ikaw na next.
So, the cells have stayed in the warm water bath long enough for the DNA to be freed from the cells. And we have removed the swab from the tube. Now, add some concentrated salt solution to your tube.
Ay, like walang concentrated solution. Joke lang. Ang micropipetter. Ba't ayaw niya?
Nakuha na siya ata.
Kumuha na ba? Ayan.
Oo, kumuha.
Ay, kumuha na pala siya
Oo.
kanina. Joke lang.
Ta.
Wow, concentrated salt. Para the salt causes proteins and other reservoirs to clump together. Ayun, para magdikit sila. Ba't sila mag-clump? Wala mo kasi wait sila mag-clump.
Diba naka-single strand pa yan sila dyan, no?
Ha? Hindi,
Single
double-stranded na yan. DNA naman
strand.
yan. Maliit lang siya,
Oh.
like, kasi di ba 2 meters daw yun. Parang
Oo.
ganyan na siya.
Mm-mm.
Tapos bakit sila mag-clump sa tingin mo?
Ha?
Bakit sila mag-clump? Baka dahil yun sa concentration gradient. Kanina. Pero ko yun. Para mag-clump
Grabe.
sila. Kasi
Na-improve
mas
yun siya isip.
dense yung solution compared sa kanila para mag-clump sila in place. The centrifuge. Place the tube into the centrifuge. Okay. In order to balance centrifuge, a tube containing water is raised up as a tube. Click the lid to turn off. Okay, sarado na natin. Ang ikot-ikot na yun siya yung centrifuge. Mix. Rowena. Hindi, sa Rowena.
Oo.
Rowena, makabasa
Ro?
ka. Feel ko lag pa rin si Ro.
Meron na.
Meron na?
Sige daw.
Sige daw, Ro.
Inside the centrifuge, the tubes spin around at high speed. The heavy clumps of protein and cellular debris to sink to the bottom of the tube, while the strands of DNA remain distributed through the liquid.
Andyan na siya. Nag-separate na yung yung proteins and debris. Like, hindi na siya like nakakalat sa solution. Bumaba na siya through the help of centrifuge.
Yan na para yung parang
Centrifuge.
bahay niya.
Use the micropipetter to carefully remove the top liquid which contains DNA and place it into the clean tube. The proteins and the other cellular debris stay behind. Okay. Yung debris kasi magstay siya sa baba pala. Tapos yung DNA yung mag-mix dun sa water. Sikos ko? Ay, hindi. Binuhat niya.
Tapos yan, etan ba yan?
Sila next magbasa. Ikaw na lang raw ulit.
Basa Ro.
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 Ito si Rowena. Or nag-lag. Ako na lang muna. Sige na. Add some isopropyl alcohol to the tube. Ay, kumuha na ba siya ng isopropyl alcohol? Kumuha na ba siya? Joke lang. Yay! Nag-clamp na sila. Ba't siguro sila nag-clamp? Inverting the tube several times mixes the isopropyl alcohol into your DNA solution. Because DNA is not soluble, does not stay dissolved in the isopropyl alcohol. It comes out of the solution. You can now see the clumped DNA with your naked eye. Sa tingin mo, X, bakit siya ganyan? Naglabas yung white ng alcohol, ng DNA sa isopropyl alcohol. Exy? Hello, guys! Naglag si Exy. Ikaw na lang, Ro. I-next na lang natin. Bigyan na lang ako konting explanation. Sa tingin ko kasi nag-separate yan siya sa alcohol, sa isopropyl alcohol yung DNA. Kasi, di man soluble ang DNA sa alcohol. So, parang nagkaroon, parang oil lang, pati water. Parang, decantation, di siya decantation. Nakalimutan ko yung process na yun, yung mag-separate ka ng different liquid. based sa density nila. Or density or pwede rin sa properties nila kasi baka organic at saka inorganic. Hindi, organic ba ang alcohol? Basta
Within the
yun.
tube separate.
Nagbasa ka raw?
Convert times.
Next na natin. Basta dito nag-separate na yung DNA sa solution with the help of isopropyl alcohol. Yay! Creation with America centrifuge. Place the tube into the centrifuge and click on the lid to close it and turn it on. This time, after the sample spins in the centrifuge, the DNA sinks to the bottom of the tube.
Parang oven pa niya.
Oven talaga. Yay!
Naman.
Okay, Ro, mabasa mo, Ro. Ro, ano si Rowena?
Andito siya. Pero wala akong marinig.
Makakuha signal siguro, no? Ikaw na lang, Exy.
Nagagalaw siya.
Nagagalaw si Rowena. Go, Rowena. We will wait for you.
Para siya nagsasalita pero wala akong marinig.
Same.
Okay lang yan, Ro.
Nag-clamp na yung DNA dun sa baba, oh. Kasi sa microcentrifuge, pati dun sa isopropyl alcohol, sineparate niya yung DNA dun sa solution.
Kaya mo yan, Rowena.
Tawang-tawa ko na talaga kay Rowena. Hindi ko makita kasi nag-present ako.
Gumagalaw talaga ulo niya para siya nagasalita kayo. Pero walang boses.
Para mga
Ayaw mag-function ng mic.
Ayun.
Ayan na.
Ayaw
Meron na, Ro.
Nawala.
Wala ulit,
Meron
Ro.
na.
Hello?
Ayun na,
na.
meron na.
Narinig na ako?
Narinig na, Ro.
Basahin ko na?
Basahin mo na.
Sige. Once the liquid is removed and the DNA is allowed to dry, you can redissolve it in the solution of your choice. You can store it in the freezer for many years, or you can move on to your next experiment. You have just extracted DNA. Try your handed DNA extraction using common household items you can find in your kitchen.
Yay! Tapos na tayo. Restart cloud na lang at saka back.